Hi, everybody. Welcome to another episode of the Labiotech Hangout. It's now the 12th episode. And so this month we have the co-founder of Stilla here. So hi, Remi. Hey. Good. It worked out. I mean, we had some technical challenges to make it work out, and good we get to finally talk live. I think we talked over emails for ages. And so you started Stilla, so life science startup based in Paris, developing next generation PCR. You are co-inventor of the technology, basically in 2010, incorporated 2013, and you just closed a Series A and a pretty big one, so 16 million. And I guess my first question will be around that. So 60 million, a lot of money for a biotech company, especially in Europe, and a lot of money for a tools company or life science tools company. So how did you manage that?
How did we raise that amount of money?
How did you manage to raise so much? Yeah.
Well, it didn't happen overnight, so it was a long process and long effort, really long effort. It was a real battle at some point. But I think so. Stilla is a company that is selling digital PCR. So it's the next generation of PCR. We've been selling our products for digital PCR. So it's a full system that we sell for 80,000 euros to customers. We've been selling our system for 2 years. We have customers worldwide. We have a few dozens of customers, all super satisfied with the product. So when we started this round, we already had basically done the proof of market, and we had traction. And I think that helped a lot in convincing investors of putting money in Stilla, because the technological risk are now, I think, fairly low in the company. And
Hello.
it's more about scaling the operations, scaling the sales. And so you need money for that, but I think it's less risky.
And then the investors you attracted, Illumina Ventures. How did you manage that, and what's their interest in the deal?
Of Illumina Ventures?
Yeah.
They
led the round, that's correct.
They led the round. And they're a US fund. So Illumina Ventures although it has the name Illumina, just something to say here up front so it's not controlled by Illumina. It's an independently managed fund that was sort of spun out of Illumina.
It's still a corporate fund. The LP is
not. Illumina is the biggest LP, but owns less than 50% of Illumina Ventures. So it's non-controlling. So it's not a corporate fund technically. It's branded as Illumina, but not controlled by Illumina. A bit of a funny thing there. So we didn't start out immediately by reaching out to big US funds. We first started working with French funds, and especially with specialized funds like Kerma, who ended up also playing a big role in this funding round. And it's through the process of fundraising, meeting with different investors, that we got in touch with Illumina Ventures, and then who took over basically in leading the round.
That's great.
It was a fairly long networking process.
That's great. I guess it took you a long time and probably almost 12 months to raise the round, I guess.
Yeah, almost.
Yeah, classic. It's great that Kerma is involved. I think they're one of the few in France/Europe who are actually investing in diagnostic and tools, and they've been pretty successful on it. So it's that's good. And I guess I had one question on what you will do on the money next step. So you mentioned commercialization and scaling up. So can you just expand a bit on it?
Yeah, so the funds will be used. Well, one of the big goals that we have for the next 2 years is scaling our business operations. So scaling production and scaling also sales. I mean, this takes money. Takes a lot of investment to go from what we are now, a 30-employee company to a 100, 200 employee company that is capable of selling hundreds of systems a year and worldwide.
So you will hire like 100 employees in the next 12 months?
Not 100 in the next 12 months, but we'll be reaching probably close to 100 employees in 12 months and 150 to 124 months. I think that's what it takes if you really want to take our current product and in its current state where we're capable of producing a few dozens a year, to a state where you can, sell hundreds, and you can do so worldwide in all major markets, so US, EU, and Asia. So yeah, big scaling challenges. So that's the main one of the main goals.
Yeah.
And then the second goal, obviously, is going to be to continue invest in R&D, bringing a second generation of our products, so new features, new products, and progressively also taking the technology, which is now for the life science research market, to some diagnostics application. That's more long term.
That's great. No, that makes, and I mean, scaling is hard enough, and it's
Yeah.
already more than a full-time job. So developing a second product on the side, it sounds already a lot. It's great. I think you went pretty broadly on everything and challenges. So now we can go a bit deeper on some of the aspects on the technology itself. So I guess digital PCR. Can you just explain a bit on how differentiate from all the PCR technologies out there, and who is your direct competitors?
So digital PCR is really the third generation of PCR. I mean, the first was endpoint PCR, second is real-time PCR, and now the third is digital.
Okay.
So what digital, I think, brings to the PCR market, it brings two things. It brings, I think, an unmatched reliability in detecting rare mutations, rare targets. And that is because, so in digital PCR, what you do is, instead of doing the PCR reaction in bulk, you start by splitting out the PCR mix in tens of thousands, even millions of subpartitions. So when you have an initially complex sample that contains, thousands of different types of DNA molecules, once you've cut that sample into tens of thousands of subpartitions, subvolumes, in each partition, you now have a much simpler sample. You only have maybe one or a few DNA molecules. And so you're performing PCR on single molecules or on low complexity samples, which allows you to get full power reactions, so full efficiency.
I guess you lower the noise in your sample, and so you have way
You're
higher.
basically lowering the noise significantly, and so you're capable of picking out one mutant out of 100,000 wild type DNA, even if that mutant is a single SNP. And if you were to do that with classic PCR, you would get false positives because you would need to amplify and amplify and amplify, and at some point you get a PCR error and you get false.
At the end, it's the big limit of PCR because you're amplifying everything. So
yeah, exactly.
And I guess the digital afterward, that's where the digital comes from. That when you have tens of thousands of samples, you need a lot of software to make sense of the data or kind of bring it together.
So the digital term actually comes it comes from the second aspect, the second thing that digital PCR brings to the table is quantification, absolute quantification. Because in practice, so with digital PCR, now the target that you're actually looking for will end up being present in only a few of your partitions in total. So you'll have negative partitions that don't contain your target. So these partitions will not be fluorescent. They will be negative, basically, zeros. And then you'll have positive partitions that contain your target that become fluorescent. And these are your ones. And so that's why it's called
count the number of positives and you can make a quantification.
Yeah. And so then in digital PCR, you transform the problem of counting molecules, which is a difficult problem because, you're counting its chemistry, and you transfer that in a problem of counting partitions, so solid objects, which in our case are droplets. So you're looking for fluorescent droplets. And that's, from a physical standpoint, it's much simpler because your fluorescence is much higher and the object is much bigger. So in our case, the droplets are 100 nanometers in diameter. Whereas the single molecule of DNA in the beginning is, it's in angstroms. And so what digital PCR brings to the table is sensitivity, what I talked about in the first part, and absolute quantification and precision in quantifying DNA. You're counting individual molecules, and that's super powerful.
And what's what's actual tech or patent that you brought into digital PCR versus others? Like,
What did we bring?
yeah, what's the key like tech
Yeah,
at Stilla?
the
Like,
key tech. So digital PCR, the funny thing about digital PCR, I mean, the concept, you can actually find the concept in the literature dating back from the early 90s. And there are already some early descriptions from the late 70s. So it's more than 30, 40 years ago that people thought about
before
this
we were
PCR.
born.
Yeah, yeah. Before I was born, people had already thought about digital PCR. About the idea of cutting down the sample essentially to a state of limit dilution, so to a state where some partitions are negative and some are positive, and then doing single molecule detection. But what was difficult in implementing digital PCR is that your PCR mix is already quite small. You're going to amplify a few microliters. And now you want to cut that up into tens of thousands, even hundreds of thousands of sub volumes, which each would be less than a nanoliter. And you want to do hundreds of thousands of PCR reactions in parallel. So that's where I come in, and that's where all the microfluidics field comes in. It really took years of research in microfluidics to get to a state where we had the technologies that allowed us to take 20 microliters, cut it up into tens of thousands of subpartitions, so droplets, and perform 20,000 PCR reactions in parallel. And so what I personally did with the people in the lab at École Polytechnique who are working in microfluidics. So we invented a bunch of tools, microfluidic tools that allowed us to do the partitioning step, the amplification, and the readouts in a single chip using a new droplet microfluidic approach. So what we brought to the field is the microfluidics.
And then you packaged it into a product that makes it more scalable and more manufacturable.
Yeah, yeah, yeah. So that's when I started the company, I had never done a PCR in my life. So 2013, never done a PCR in my life, but I knew that I had the microfluidic tools that allowed me to take 20 microliters and cut it up into 20,000 1 nanoliter droplets and potentially do a PCR reaction in those 20,000 droplets easily.
And
And it turned out to be true.
I guess in 2010 you basically had the basic technology, and then you looked for application instead of taking the opposite approach. And then
Yeah,
it ended
yeah,
up PCR being the perfect candidate, and you were
yeah.
pretty right.
Yeah, basically. So yeah, I did my PhD between 2009 2012. I mean, the core tech, the two core patents were filed in 2010, but then still it took a few 2 years to mature the technology, have some prototypes that worked well to partition samples easily, and in parallel, while we were trying to make the technology more robust, yeah, I was on the lookout for other approaches, other applications, and I learned about digital PCR, and I was immediately attracted to this technology, which I think is super smart. It's a super smart concept to do gene detection, or I mean DNA quantification and detection.
Yeah, definitely.
Uh,
And so on digital PCR, so I guess it will replace all the existing PCR technologies, at least in a clinical setting or at scale. And so
Maybe
who are you competing with on the digital, like purely on digital PCR?
so purely on digital PCR. So the pioneers that really invested in the technology first were Fluidigm, who launched the first product in 2006. So they were the pioneers, but their technology ended up being really too costly. So now they're mostly out of the market. They were replaced as leader in the market by Biorad. So Biorad is the current market leader. Biorad acquired another microfluidics company called Quantalife in 2011, and that's how they got in the digital PCR field and have been leading since then. Then Thermo also has a product. There are a few small companies that are developing or selling digital PCR solutions on the market right now. But it's a super fast growing market. It went basically from 0 million market in 2011 to several hundreds of thousands of million euros in now, so in 6 years.
And I guess the growth rate, yeah. So and it will continue in the next 10 years.
But it's not going to replace. I don't think it's going to fully replace qPCR, because qPCR or real-time PCR works well for some applications,
And
and
it's much
it's still
cheaper
less
to do,
expensive.
so for some some application, it will still be relevant.
Yeah. So digital PCR is more expensive than qPCR. So I think right now digital PCR is replacing qPCR in high-value applications, or where in any application, at least where you need to quantify DNA. I think it makes obvious sense that digital PCR is superior to quantitative PCR, and that's the main niche that we're targeting, and that I think globally digital PCR is targeting.
No, that's great. So I think we talked enough as a technology. If we go to the application side of things, like you list on the website, I saw and you listed a few applications, which is pretty general. And I guess you're really a platform that can be applied everywhere. But what's the main application right now?
Yeah, so we're a platform solution, we're a technology provider, and we don't verticalize on a specific application. So people can do GMO detection or quality control in pharma or liquid biopsy analysis with our system. But what I think
What's
what is really
what's a
exciting,
major one for you? Like
yeah, the
you
major
must have
one I
one
think
segment where you have more usage.
it would be in the field of oncology and in the field of liquid biopsy analysis. Because this is really an application where digital PCR makes full sense. If you take a blood sample and you're trying to detect tumor DNA in a blood sample, you're really looking for a needle in a haystack. You have a ton of wild-type DNA, which is human DNA, human genomic DNA. It's super close to the mutant DNA. It's really, a single SNP that differentiates the mutated DNA from the wild type. And so you're trying to find a close relative to wild-type DNA, but at very low amounts, and you want to quantify that as well.
Yeah.
And digital PCR is the only solution that allows you to detect a few of those tumor DNA molecules in a ml of blood and to do so reliably. And I think right now there are some really exciting applications where we're capable of detecting the resistance mutation to some therapies from the blood of a patient, and you can monitor that as well in time. So physicians are using digital PCR assays on cancer patients to monitor if during treatment some resistance mutation appears. And if it does, then there's some early clinical data that shows that it really means that you should switch drug because the cancer has evolved in a resistant cancer type.
That makes sense, and it makes me think directly. I guess Grail is using your platform.
No, so I think Grail, they're really focused on sequencing, and sequencing would be the other. On the others, on one end of the spectrum, you have qPCR or even PCR, and on the other end, you have sequencing. I think the Grail effort is super interesting for us as well, because Grail is really trying to find everything that you can find about some of cancer patients' cohorts. They're really doing an extensive search of what's clinically relevant for cancer types. But my honest belief is that at the end of all the studies they're making, it will come down to a few biomarkers. And the relevant clinical tests to be implemented, routinely to tens of thousands, millions of patients won't be a sequencing run that looks at everything. It will be either targeted sequencing if you still need to look at a few dozens of targets. But if it's only a few handful of targets that you need to find, then digital PCR will probably be the clinical tool.
And why can't you just combine both? Like
combine?
Yeah.
You do. Because I think sequencing makes sense in the beginning when you're trying to understand what you need to look for a given cancer patient. But once you know what is the disease and what are the relevant biomarkers, then to monitor, I think that a targeted approach like digital PCR is much more cost efficient than sequencing. So it's combining the two technologies.
At the end, if you look at looking at the cancer patients, you would start with the sequencing to understand better what's happening, and you will also want to quantify over time, and so you would have like sequencing and digital PCR working side by side.
Yeah, that's I think a likely outcome of all the efforts that are being done by Grail, by others, by us.
Yeah.
I
No, it's
really don't
great.
see sequencing as a competitor. I think we will need to work
That's great.
with sequencing.
And that's great. And on what you said about verticalizing, I think it makes total sense to be a platform if you can be a platform.
Mm-hmm.
But it makes also sense to verticalize if you see one vertical or one segment that explodes, typically liquid biopsy, which probably has some specific needs compared to keeping it horizontally. So can you expand a bit on why you chose to stay horizontally versus going into one vertical?
Well, I think for us it's a luxury not to have to verticalize because it reduces risk. I mean, as soon if you're a company, a startup company, and you have a new technology and you have to point one or two key applications and you have to make those two bets conjointly,
Yeah.
the probabilities of conjoint success drops fairly low.
And your probability of success as a startup goes even lower.
Yeah, super low. And so for us, we're lucky to have a system that's it's the same product for any type of application, and so we can build a market, we can build revenues without verticalizing, and so we can have this more cautious approach of waiting to see really what is the most promising application and to verticalize maybe later at a cost. I mean, if we do so, we might need to acquire a company that has developed an interesting application and do so at a cost higher than if we have developed the application ourselves. But then it's less risky. And so if you think about it, it's exactly what Illumina has done.
Yeah.
Illumina
It was
waited until you had those companies doing prenatal diagnostics, and then they bought one when they were sure that it would be a huge market for them.
Yeah, and still the
So
Illumina machines are still like really a platform and not that specialized when you.
yeah.
Okay.
So we're basically waiting, seeing. We do have some efforts. We're still investing R&D, having collaborations here and there to develop applications to mature them. But yeah, we want to stay a technology provider at least for the next.
And I guess you have good advisors at Illumina now to know how to do it properly.
Yeah, I guess they can help us with that as well.
All right, that's good. I think we're running a bit out of time, but you talked about traction in at the beginning. I think like that you have already several clients. I think we can pass on that. I have one question that you didn't talk too much about was on the software side of things. So I think you have your own software, and I was curious on like kind of why do you need your own software? How much software do you need? Can't you use any existing tools? Why is there not any other like provider of software? Like yeah.
Yeah, so software is actually where we invested quite a lot in R&D. We have a full software team, and actually, so if you think about our products, we reuse like our instruments. We reused a lot of existing tools to make our instruments, but software we really had to build from scratch because the data we get out of our technology is so different from what exists. It's basically a big image on which you have to locate tens of thousands of droplets. So there's some image analysis algorithms. We use AI, neural networks to do that. And then the data analysis for digital PCR is quite different. It's close to cytometry, but it's not cytometry. So we really chose to invest in R&D and to have a software in the end that is built to be user-friendly to our customers and to be 100% aligned with the goal of the product, and not something that is reused but not ideal, because we really believe strongly in ease of use and in taking into account the user experience, even for life science tools. We're not a software app, but
that's good. I hear it more and more from life science tool startups or companies who are really, I mean, developing more and more into their own software. First, because it adds a lot of value for the end user, and second, because the existing software is pretty crap.
I think that's pretty true. I think it was something that was really missing in life science tools even like 5 years ago. I mean, software were really, really crap, and it's not hard to provide better things than what already exists.
I remember when I was working in a lab, I was like, guys, like that's insane to offer that. Like,
LabVIEW software, it's like what?
yeah, like
No,
it's so
we can
hard.
we can build tools in C, C++, not LabVIEW.
Yeah, even the
Sorry.
intern can do way better, like in just in a month. Like, yeah. And and I guess I had a question on. It's good. It's a perfect transition. What's missing in the life science tool space? I think software answered. Anything else you see, like, or like, what's your from a bigger perspective, not just digital PCR, more like
Yeah.
broader, like what's missing?
I mean, there's one field that has my interest right now is sample prep and sample extraction tools. Because so if you think about liquid biopsies, we now know that like all the information about a patient is present in a 10 ml of blood with some rare biomarkers. But the challenge is not now in finding those biomarkers once they're in your analytical system. Like if the DNA molecule is in our chip, we'll find it, we'll detect it. But the challenge is how do you extract that DNA molecule in 10 ml and make sure that it ends up in the 20 microliters that you analyze with your machine. Um,
If it doesn't get lost on the way, or like,
it doesn't get lost on the way, and it's a sampling problem. If you have one molecule in 10 ml, but you only look at 20 microliters out of that 10 ml, I mean you won't find that one molecule of
yeah.
interest. And I think that's a challenge that has been overlooked. That sampling challenge. What you need to find is often in low quantities, and if you really want to find it, you have to concentrate highly what you want to find. And that's where I think sample prep, sample extraction tools can be helpful, but maybe not
And I
as expensive.
guess you will have some microfluidics involved.
Maybe, and you could even think, to have some inline tools that you could just get the blood passing through a chip, directly plug through a catheter in the patients and have filter out multiple mLs of blood and make sure that you're capturing what you need to detect on the chip or on the device, the inline device, and then get what's extracted into your analytical tools. I think that's a missing piece of the life science approach right now.
That's good, and I guess would help you a lot then.
Yeah, it would be upstream of what we do. But I think now, I mean, if you think about life science and genetics, I mean, genetic analysis, we're getting close to perfection in terms of the tools we have.
Hmm.
If what you're looking for is what is in your sample, you'll find it. But now you have to make sure that it goes into your
I
system.
guess you still have a cost issue, and you probably need to lower the. I mean, the cost of detecting will probably need to be lowered by a factor of, I don't know,
Yeah,
five
yeah,
to
you still
ten.
need to
But
improve on cost, but I think the
the
technology,
product is here.
sequencing technology, digital PCR, are getting really advanced.
Yeah.
And if you think about what's less advanced, it's probably upstream.
Yeah, that's cool. And I guess just switching gears to the last question on the team, you mentioned you would hire a lot. So I guess how and connected to what we discussed, that you invest in software, your own software team, and pumping up sales. So like, what's the focus of who you will hire, and will you hire everyone in France, or will you hire open other offices? Like, what's the yeah.
So we have a big effort. I think this is since it's Labiotech.eu, then I might talk about Europe, but we'll have a very strong effort in scaling our sales team in Europe. So we'll be hiring sales reps, application specialists, support, field service engineers, marketing managers, I think, in Europe. That would be overall Europe, having sales reps and field and application specialists in UK, in Germany, Nordics, France, obviously,
A
Benelux.
remote team managed from Paris or headquartered in, I mean, was
Yeah, yeah, yeah. Obviously, with them coming in
yeah.
at their headquarters often, but yeah, we will soon. I think by the end of next year, we'll have a global presence in Europe with with employees on the ground.
Okay. So how can they apply if they're watching us right now?
Yeah, they can apply. They can send us their CVs. We're definitely looking for high profiles in sales and applications specialists.
And I guess it's pretty rare. I mean, it's not that easy to find across Europe. Good specialists and good sales rep. It's
not super easy.
All right, that's great. Great, and I guess next milestone we mentioned already. So I think the last question is just who should we interview next?
Who should you interview next?
young biotech entrepreneur under 35 in Europe.
Did you do DNA script already?
Not yet.
Oh, man, I think that's a good one.
Okay.
Either Sylvain or
list.
Thomas.
Yeah.
Yeah.
Another one.
Made some super progress recently.
Yeah.
So definitely one company to look at.
Cool. I will do that. All right, great. Thanks a lot, Remy, and
thanks, Philippe.
Glad it worked out, and glad
it wasn't easy.
your phone hold the whole time, and I think it all worked fine. So, yeah, have a nice day, and
yeah, bye.
see you around. Bye bye.
Thanks a lot. Bye.